First, I'd like to thank the organizers for this invitation. I'm really happy to be here. I think it's a really exciting workshop. And I feel like I'm learning a lot listening to these presentations. And now I will talk about embryonic development, and more specifically about embryonic morphogenesis. And a lot of data I will show you has been acquired actually at Caltech. In concerning the visualization tools we are using in developmental biology, I would say, as shown in the program, actually, we are sitting in between, let's say, cell biology and medical imaging, and neuroscience is not too far away. So in some aspect, it's useful because we can use the same kind of tools in terms of microscopy, imaging, software. In some other aspects, it's not so good because as we are, I would say, a small community, we don't have the tools that are really fully adapted for what we are doing. So concerning the link with medical imaging, we can use the same kind of microscopic imaging techniques such as magnetic resonance imaging. So I show you here some data looking at quail. This is data from the digital three-dimensional atlas of the quail development. And I think in this specific case, the challenges concerning visualization are really shared with medical imaging. It's, for instance, the nested object problem, the fact that we have several layers of tissue here, and if we want to visualize the different tissue at once, we need to use a specific transparency or specific way to visualize the data. Nice thing here is that we can go into the egg, for instance, here of this quail, looking at every structure inside the embryo. This embryo is quite big. It's a few centimeters large. If we go to smaller embryos, smaller species, we can use other technique of imaging, such as microscopy, and then we are closer to cell biology. So here, this is imaging of a fly embryo, part of the embryo during gastrulation, with imaging of fluorescent imaging of the nuclei, and this is really close to the presentation we had yesterday about cell biology. We use techniques that are the same as in cell biology, such as fast spinning disc confocal microscopy here to image this. This is a 3D stack. So yeah, the data, when we talk about visualization of biological data, the data we are using are images. And from this, we can extract things like the position of the cell using 3D segmentation, such as in cell biology. The difference is that in our case, most of the data are three-dimensional, or even more dimension if you include the time, so 4-dimension. And from this segmentation of the nuclei here, you can follow the cell movements and track the cells. But the fact that we are mostly working with three-dimensional data is quite challenging in terms of visualization. In the previous case, the cells were organized in a monolayer, so it's quite easy to follow. And in many cases in developmental biology, people are focusing on 2D morphogenetic processes just because it's hard to visualize if you look at 3D morphogenetic processes. So when you, for instance, I show you here an example of tissue morphogenesis, and when you look at a tissue, it can be seen as a 2D process. And here I show you the example of cell intercalation occurring inside the tissue of the fly during gastrulation. I don't know if you can see well, but let's go back here. You will see the cells here will intercalate in between these two cells here. This process is also occurring in 3 dimensions, but it's much harder to visualize. So here this is cell intercalation occurring in 3 dimensions into a zebrafish embryo with labeling of nuclei again here, and you see the tissue, and you have actually a lot of cell intercalation occurring here, and it's really hard to just visualize if you look at the raw data. You can see it just looking at the raw data here. So if you do some processing, you can track a few nuclei and then focus on a small amount of cells, and then you can really highlight cell intercalation. So for instance, here, this cells will intercalate in between the gray cell and form a kind of tube inside the embryo. And you will see at the end, well, the tube formation here. So you form a kind of monolayer, and the cell intercalate in 3 dimensions to form this tube. But this is actually unpublished data we took at Caltech, and it was really hard to work with this, and we were all busy with other things, so we just decided to switch to something else because it was too hard to work with. So I think it's the case for more than 90% of the project different biologists are working with. I think at some point the challenges are so high that they just give up and just try another strategy. So that's why I think the challenges and the needs for visualization tool in developmental biology, it's really hard to see them in the literature because most of the challenges are behind the scene. So that's why today I will try to focus on a few things that are not, you cannot see in the papers, in the final work. So another aspect is that these days it's really exciting to work in developmental biology because there is an explosion of fluorescent techniques that are really adapted for embryo development. So I will not talk about precisely about the microscopy technique, but this is a different kind of fluorescent microscopy technique. I will actually show you, I already show you actually different technique used for imaging embryos, such as confocal spinning disk, light sheet microscopy. I actually recommend you to check one of the posters on SPIM imaging, or two-photon microscopy, or we actually developed at Caltech the two-photon light sheet microscopy. So I will show you data using these different techniques. The thing is that we can go deeper, faster, with lower phototoxicity using the adapted technique of microscopy, and we generate a huge amount of data, of multidimensional data. And the challenge is to visualize this data, to process this data, to visualize the features we are computing on this data. And I just realized that trying to work on this presentation, that visualization is an issue for almost every aspect of my work, from the time we are acquiring the data to the time we are publishing the paper or communicating the work. So I show you here, for instance, some data from EMBL using SPIM to image development of a zebrafish embryo. This is the first 24 hours. You can image thousands of cells. So here, this is 2 different views. The thing is that here you can quite easily follow the development because most of the development is occurring at the surface of the embryo. Just by visualizing these 2 views, you have an idea of what's going on. But if you go later into the development, or if you look at other embryos like fly, for instance, quickly the movements are in 3 dimensions. It's really hard to follow just by looking at the raw data like this. Show you here the use of two-photon light sheet microscopy to image deep into highly scattering tissue like a fly embryo. And here we have also labeling of nuclei, and this is the anterior development of the embryo until the embryo is hatching at the end of the movie. And here we have a cellular resolution, so we can basically track the morphogenesis of the anterior development. So we start to, we are going toward high throughput and high content imaging and analysis, and it actually raised a lot of visualization issue. If we are inspired by what is done in cell biology, we can follow the same kind of workflow for analyzing this data, from the imaging acquisition to the analysis using data registration, segmentation, like nuclei, as I showed you, feature computation. Some of the features are really specific to morphogenesis, data mining, and modeling and simulation. And I would say in every of this aspect, maybe excepting the first one, visualization is an issue. The visualization of all this multidimensional data. So I will maybe focus on a specific project I work on because it's easier for me to know what was going on behind the scene. So let's focus on fly embryos, nuclear labeling, and we look at gastrulation process. And the first process of gastrulation is this invagination of the ventral fluor here. So this is using a commercial microscope here, a two-photon microscope. we look at the entire embryo, and then we can track the cell movement. So this is a representation of the trajectories of the cells. The color code for the time here, so it's blue when it's at the beginning and yellow at the end of the process. And what you start to see here is that it's a big mess. It's really hard to understand what's going on, especially when you are not familiar with the process. So we need to find a way, if we want to understand the morphologic processes, to analyze this data, to visualize the features we want to compute. So we were specifically interested into the movement of the mesoderm cells that are inside the embryo here, forming this tube. And after the formation of the tube, the cells are spreading. These cells that are the mesoderm cells are spreading on top of another layer that is the ectoderm. I don't know if you can see well here, but this is basically the process. You have a tube of cells, and the cells will collapse, the tube will collapse, and the cells will migrate on top of this outer layer here, the ectoderm. So we imaged this movement. We imaged both the ectoderm layer and the mesoderm layer, and it's actually really hard to show you the raw data because we have cells everywhere. So I show you here a visualization that helps to understand the process. I just display the raw data for the ectoderm layer. So from this view, it's easier to see. And I just selected the track of a few mesoderm cells just to show you the spreading on top of the ectoderm layer. I just remove the mesoderm signal here. And then you can track, you can show the tracking here. So in gray, it's the ectoderm layer. If you are familiar with this, you can see the movement of convergence extension of this layer. And then on top of that, you have the spreading of the mesoderm cells. So here, one visualization challenge is to simultaneously display the raw data, the raw imaging data, and the process data at the same time. So then we wanted to focus on the specific movement of these mesoderm cells, and something really important was to decompose the movement in something that is meaningful in terms of the biology. So we choose to use a coordinate system that was adapted to the body plan, and we were lucky enough that this embryo in this area has a cylindrical shape. So we could use a cylindrical coordinate system, and we could decompose the two-dimensional movement into each coordinate system and display the data instead of showing the three-dimensional data, display the data this way with time in this direction and each coordinate system in the other. And each line here corresponds to the track of the cells. And it's really nice here because you see each direction corresponds to a specific process. You have in the z direction, this is the main axis of the embryo. Here, the anterior-posterior axis, you have the movement of convergence of the jambon in this direction. In the radial direction, so from the inside to the outside, this direction is also the apical-basal direction of the ectoderm layer. In this direction, you have the collapse of the flow. And then in the angular direction, which is basically the left-right direction here of the body, you can see the spreading of the cells. So then, once we had this decomposition, we could play really with data, and we could, for instance, unwrap the ectoderm layer. This layer can be seen as a tissue layer, so you can see it as a 2D plan. And then you can look at the mesoderm movement on top of this layer. And you can display the data this way. So you can see really well in gray the movement of convergence extension here. And then on top of that, I just selected a few mesoderm cells to show you the movement of spreading. And this was really important for us during the process of analyzing data, because at the beginning it was really hard for us to understand what was going on and to compare Y-type with mutant embryo. And looking at the data this way, it was much more easy to understand. And one thing that is really obvious here is that you have a clear correlation in the movement in this anterior-posterior direction here between the gray and the orange cells. And we could actually quantify this and demonstrate that the mechanical coupling between these two layers of cells in this anterior-posterior direction. So in practice, it means that it's like the mesoderm cells are moving on a moving walkway. So the movement is actually the composition of two different movements, the movement of the ectoderm layer and the active movement of the mesoderm cells. So in practice, it means that we could process the data to stop the movement of the ectoderm, just to look at the residual movement of the mesoderm cells. And also, if there is no more active movement in this direction of the mesoderm cells, we can basically forget one dimension. We can forget this anterior-posterior movement. So it means that in practice, we can limit our analysis to only 2 dimensions instead of 3 dimensions, only the radial and the angular direction. And this was basically, we realized this because of the visualization, because of that kind of visualization we did here. And then what we did here was just to represent the mesoderm cells movement before processing, so from the raw data. And then in blue here, it's the movement of the mesoderm cells when we artificially stop the movement of the ectoderm layer. So you see that you don't have any more movement in the anterior-posterior direction. So here, one visualization aspect was to show on the same image both the processed data and the raw experimental data. Okay, so I'm not sure that it's, when we try to publish this and we try to publish protocols of our experiments, actually, for us, at the end, when you spend so many hours looking at your data, you have all the movement in your mind in 3 dimensions, and you can easily talk about it. But when you present the data in 5 minutes, it's really hard to understand what's going on in all these dimensions. So when we try to publish this, especially the protocol, the editors ask us to make an animation to explain the different movement in our experiments. And actually, it was quite challenging for us to try to explain this. And this is the end result. So this is the only movie with artificial data, actually, one animation that we did to explain the different body plan, the way we acquire the data on the ventral side here, the mesoderm of the embryo, and then the formation of this mesoderm tube here, and this ectoderm layer, and then the synodical coordinate system adapted to this body plan, and then the movement of the mesoderm cell compared to the ectoderm layer. So I don't know if this, I hope this animation helped you to understand the movement in 3 dimensions. But this was also a challenging issue in terms of visualization for us to make that kind of animation. Okay, so let's go back to the data. So we wanted to check many different aspects of morphogenesis, and one of the aspects is the cell division pattern. And when you look at the raw data here, so this is just one plane. It's really hard I cannot show you the 3 dimensions because you cannot see anything. So this is just one plane. You have the anterior-posterior axis, the spreading of the mesoderm cells, the ectoderm layer, and you see a lot of cell division everywhere. So our first question was to ask, are every cell dividing and how many times? And then to do that, we actually try different ways to represent the data, to visualize the data. And, so you cannot see, so you cannot see much. You cannot see so here this is the representation of the track. This is the angular direction, this is time, and you should be able to see some track behind here in gray, but you cannot see them. But you can imagine that you have a lot of tracks everywhere here. This is the cell tracking. And on top of that, I represented 1 point for one cell division. And we realized that there was this really nice wave of cell division and 2 waves actually of cell division. And then if you use a color code to code the position of the cell at the beginning of the process, so within the tube, before the tube collapses, this is the radial direction. So the red cells are the cells coming from the outer part, and the blue cells are coming from the inside of the embryo. It's like the cells are keeping the memory or where they are coming from. So depending on where they come from, they divide at different times. And there is no way you can see that by just looking at the raw data. First, because you cannot see any specific wave of division, and also you don't have the memory of where the cells come from within the tube. in 3 dimensions. So something really interesting for me here was to realize that we try different ways to visualize the data, and then this visualization reveals new insight we didn't expect it. This waves of division is not specific to fly. If we go back to the SPIM data of the zebrafish, in this paper done at EMBL, they look at also wave of division, and they have a really nice way to represent the cell division. They track the cells, and they display in red the cell just before division, and in blue the daughter cells. And if you look at the first division, you can see waves of division. But the difference is that here, if you go back to the raw data and imaging data, you can actually, if you look carefully, you can see this wave already here. It's also because at the beginning it's really at the surface of the embryo, and you don't have too many cells, so you can actually see the waves from the raw data. So in this case, in this specific case, the visualization tool is to highlight specific processes that we already know. We just want to highlight this way of division. It's really nice. It's a really nice way to show it. But we are not learning much in terms of the scientific point of view. We already saw this wave from the raw data. I just wanted to point out here that the difference here is that only during this visualization that we can identify such pattern of cell division. Okay, so if we continue on this Mesoderm project, we look at also the pattern of migration. And I show you here again, the angular value here, the time here, and the track here. And we use a color code to code the angular position at the beginning. So it's the angular position within the furrow here. And what you see here is that you have a really nice organization that is kept during the whole process of migration. And then we could display, using this color code, the data in a time lapse with the angular position here, the radial position here. You have the cells that are collapsing here and then forming this monolayer at the surface of the ectoderm. And then you can compare this with mutant. So I won't show you much about the mutant, but the problem with this is that we had in terms of visualization is to display on the same graph several wild-type embryos and several mutant embryos, and to compare the two. So we decided to go to something easier to display. Instead of displaying the whole graph like this, we just look at the position at the beginning and the position at the end. So, and we display this with this graph here. So this is the position at the beginning, position at the end, and each point corresponds to one cell. And depending where you are in this graph, you have different behaviors. So it's hard to see because it should be white, it should be gray, light gray here, and dark gray here. So if you are in this part of the graph here, then this is the normal behavior. You start here, the cells spread and go in this position. If you are here, it's actually wrong. It's going toward the midline. And if you are here, the cells are actually going on the opposite side of the embryo. So it's completely disrupted. And then when you show the wild-type data, it's organizing like this. Every cells are in this part of the graph, showing the right movement. And then from that kind of graph, you can display several embryos on the same graph, and then show that the behavior is the same. And then you can show mutant embryos. So in terms of visualization, here the issue was to manage to find a way to display all the data on a single graph. And I won't go into the biology insights, but it was really useful to do that for us. Yeah. Displaying simultaneously data from different experiments is really also, an issue for other application. And here, this is back to zebrafish, looking at this cell division pattern. And in this paper, they recorded the data with segmentation of the membrane here for different embryos, and they reconstructed the pattern of cell lineage in space for the 6 embryos together. And they reconstructed this prototypical zebrafish embryo, starting with one cell at the beginning, and they show the average orientation of each division at the beginning of the development. So it's really nice. Here you can display, you can combine the data from different experiments on that kind of graph, but it's limited in terms of how many cells you can follow, because it's here at the end we have 128 cells, but if we have only 7 cycles of cell division, and the embryo will quickly reach thousands of cells, so it will not work for later stages. So visualization, the data from multiple experiments, especially when you start with multidimensional data, is really an issue. Okay, so just the last project I wanted to talk about is, again, looking at zebrafish, but a completely different process, looking at the left-right symmetry breaking in this embryo. And at this stage of development, 12 hours after the beginning of the development, the embryo looked completely symmetrical from the left-right aspect. And you just have this small vesicle called the Kupfer vesicle, where you have a flow that is generated inside that is breaking the left-right symmetry. And I wanted to show you here that visualization aspect was really important for different reasons. So this is an imaging of the Kupfer vesicle using two-photon microscopy. You cannot see the flow here because there were no labeling of the flow. And at the surface of this vesicle, you have a lot of cilia that are beating, and you cannot see them here because they are beating too fast. We could find a way to seed the flow with a fluorescent particle and to track this particle and to display the flow within the vesicle. So here you see this is the anterior-posterior axis, this is the left-right axis, and from this data set here we display the flow that is actually breaking a left-right symmetry. So the question was how to generate this flow, how the cilia are generating this flow within the Kupfer vesicle, and how you break the left-right symmetry. So we know that the vesicle is full of cilia at the surface, and we wanted to know how to generate this, how this cilia can generate the flow. So it's known that you have different kind of cilia with different kind of beating patterns, and what we know is that at this, so this is some basic hydrodynamic aspect. We know that in these cases, to generate a flow, we need to have an asymmetric beating patterns at the scale of the cilium. And so this is a pattern that are known, but we know this, in this case is the cilium bent with an asymmetric pattern. And we know that in most, I mean, at least in mouse and also in zebrafish, the cilia are not bending. They are stiff and they rotate like this. So there is actually a third case here, which is probably the case for every left-right organizer. And in this case, we break the asymmetry at the scale of the cilium by tilting the angular, the axis of the rotation. So you have a cilium that is beating here, and you have a tilted axis in a specific direction. And when you tilt this angle, you will generate a flow that is perpendicular to this direction. So the idea here is that the angular direction of the axis of rotation is really critical, and we wanted to see it and to measure it. The problem is that it's really hard to image the cilia because they are beating really fast at 30 Hertz. So it's almost impossible to image in 3 dimensions using a fluorescent microscopy technique the beating pattern of this cilia. So what we did was to try to use visualization to overcome this limitation. And what we knew, what we were able to do was to label the flow surrounding the cilia. So we just look at the flow surrounding close to the surface, and we could identify some vertical pattern of flow. So this is a visualization of the flow of the particle that are trapped within the flow close to the. Oh. Maybe I'm late. So it was the last slide. No. it should work. I think it's maybe the projector, no?
It's not good. Oh, yeah. You might have hit
Hit the black button. That's
on the.
yeah, there you go. That's the
No.
blackout button.
Oh.
You hit that. Yeah, that makes the screen go black. That's forward and back.
Okay,
Just
okay.
don't hit that. Oh, you're gonna hit it again. And then wait for it to go. Still trying to push the video up. There you go.
Okay, sorry. Almost the last slide. Okay, so I was here. So we could display this and interact with this data to actually draw a volume inside this flow to identify the axis of rotation of the cilia, and then we could identify the position of the cilium, and specifically looking at the angle, the angular aspect. So I wanted to show you this because visualization was really critical in this case to basically overcome the imaging technique limitation. We couldn't see the cilia themselves, but we could image the flow surrounding it, we could display it, and we could interact with data to reconstruct the position of the cilia and make some measurements. And then we could start with this. This is the raw data here with the particle, and then this is the surface of the cells, and you could reconstruct the position of the cilium and then the flow surrounding it. Okay, so my last slide is a list of all the challenges I could think about. I think it's really not exhaustive, but I don't know if I will go through everything, but I try to illustrate a few of these points. For instance, one big challenge, I think, concerning visualization in developmental biology is the interdisciplinary aspect, and it's actually really hard to bring together developmental biologists with computer scientists. For instance, I don't know many places where you have a computer science lab in the same building as a developmental biology lab, and that's a shame. Also, I would say the imaging people in developmental biology, it's a quite small community, so we are basically using commercial software that were designed for other disciplines like neuroscience, cell biology, or medical imaging. In some cases it's good, in some other it's not, because it's not specifically designed for what we are doing. Also, I think most visualization challenges are behind the scene. It's not published in the paper, so it's really hard to understand the needs just by reading papers. One of the main challenges is the fact that we are dealing with highly multidimensional data. We really need to remove background information from the relevant information to be able to work with this data. We also would be really nice to be able to interact with the data that are both the raw data, the process data, the model and simulation at the same time on the same graphs. Okay, and then we also going toward high throughput and high content experiments, so it means that we are dealing with a lot of data, and visualization is really critical. And why should we improve the visualization tools? First, can reduce the apparent complexity. I wanted to really point out this because it's not because it's hard to see or because you have a large data set that it's complex. Maybe by using the right visualization tools, you realize that it's actually simple. And it was actually the case for the fly embryo imaging. At the beginning, we started with all this track in 3 dimensions. It was really hard to understand. And we realized that everything was highly organized and really settled, and at the end, quite easy to understand. And the problem is, it's a bit frustrating because when you show the result, the result to people, they don't realize the work that is behind, because at the end it looked straightforward. So that can be a bit frustrating. Okay, so thanks for your attention. Just wanted to acknowledge people I work with, like Tai Trung, Amy McMahon, Angela Statopoulos, Scott Fraser, Julien Vermeau on the Physical project, Michael Liebling, and the whole Fraser lab at Caltech. Thanks.